All right. So first of all, I want to say that I'm really honored to have been invited to speak here today, especially to honor Bob, and especially because I myself, as you'll quickly learn, don't work on tough cells, and I don't really work on taste receptors either. So it's especially an honor to have the opportunity to learn from all of you and to be selected to speak in this event. So thank you for that. I also just wanted to say that when I saw the list of speakers, I knew that at this event I would learn a lot. I knew that I would learn a lot about tough cells, and I knew I would learn a lot about taste receptors. What I didn't anticipate is that during the coffee breaks, I would also have the chance to learn a lot about large North American mammals. That's actually been really fun, and I got to say this is a really, really cool venue for a talk. So thanks to the organizers for that. So as has already been stated, I study olfactory receptors in the kidney. And I know everybody here already knows a lot about G protein coupled receptors, but I kind of want to introduce this gene family to you from the point of view of how my lab thinks about them. So, of course, we know that G protein coupled receptors are the receptors that sit on the surface of our cells and get information from the outside world to tell our cells what are going on. And this information can come in the form of a bunch of different types of signals, like neurotransmitters or hormones, or even things like light and odors, or I should have added taste sense on this slide as well. One reason that we got really interested in GPCRs is because they're the most druggable gene target that we have. So there are more FDA-approved drugs that target G protein coupled receptors than any other type of protein. And I'm a renal and a cardiovascular physiologist. And so I put on the bottom of this slide 3 really famous GPCRs in the space that I work in, and 3 really famous clinically relevant antagonists that are used in the clinic pretty often, just to kind of remind us that these receptors are druggable, they're targeted in the clinic, and this can be very efficacious. Now, our lab is a little bit weird in that we're not interested in these super famous receptors like the vasopressin receptor, the angiotensin II. Instead, we're fascinated by the fact that there's a large number of renal GPCRs that don't have known physiological roles. So if you look up something like the angiotensin II or the vasopressin receptor in PubMed, you'll find hundreds and hundreds of papers telling you all about it. But there's a large number of renal GPCRs that, at least on the RNA level, some of which are expressed at a pretty similar level to these famous ones. And when you look them up in PubMed to find out what they do in the kidney, you can find 0 papers. So we're really fascinated by that. And we think that at least some of those understudied or unstudied receptors might do important things just like the famous ones do. And so we kind of want to explore that and try to look at them and try to begin to understand that. Now, a large number of these understudied GPCRs are olfactory receptors. So I know everybody here, again, is familiar with olfactory receptors. They're a really common gene family. I'll just remind you that they're the largest gene family in the entire genome. So you have more genes in your DNA that encode olfactory receptors than encode anything else. But a lot of them don't have known ligands. We know we can smell lots of things, and we know that we have lots of receptors, but we often don't know which receptor is smelling which thing. Of course, olfactory receptors play important roles in the nose, where they mediate our sense of smell. And in recent years, it's become clear that they also play important roles in a lot of other places. And I'll say taste receptors too, right? I'm not going to talk about taste receptors today, but I think that the speakers coming before me have made a beautiful case for the fact that taste receptors play really important roles in lots of other places. In my lab, we're particularly interested for a role of olfactory receptors in the kidney. I was trained to think of the kidney as a master regulator of homeostasis for the whole body. So the kidney's job is to monitor so many different things and make sure that all of those different parameters stay within a homeostatic boundary. And if your job is to be the master regulator of homeostasis, I think it makes sense that you might have a good reason to make use of sensory receptors to keep track of so many different parameters that you're supposed to be monitoring. So a key question that drives work in our lab is, what role do olfactory receptors play in kidney physiology? And a little bit uniquely, I think the way my brain works is I'm always most interested in the physiology, even more than the pathophysiology. I really feel like for so many of these receptors, we don't know anything about what they do. And we need to understand what they do in health first before we can try to understand how that goes wrong in disease. So you'll kind of see my own personal bent come through, hopefully, as I speak. So the purpose of today's talk, I'm going to kind of talk about two different categories. So the kidney is made up of a renal tubule, also known as a nephron, which is a polarized epithelium that has specialized transporters in different segments. But there's also really important roles for the vasculature in the kidney. There's some very specialized vessels that interact with this tubule in very specialized ways. So I'm going to tell you first a short story about a novel GPCR, an olfactory receptor, actually, in the renal tubule. And then I'm going to spend the majority of my time talking about a new story we're particularly excited about in the renal vasculature. But since I don't have time to tell you, about lots of different GPCRs in the renal tubule, I kind of wanted to summarize a general theme that's come out of our work on the renal tubule. And that general theme is that receptors in the renal tubule, the ones that we've studied to date, all seem to modulate either the activity, the expression, or the localization of transporters, which are already known to be famously important in renal physiology. So on this slide, I've listed on the left-hand side several different GPCRs that we've studied, including an olfactory receptor, the place that they're located. So those are very different cell types within the kidney, such as proximal tubule or intercalated cells or principal cells. And then in each of those cell types, the receptor that we've studied seems to be modulating either the activity or the expression of a classically important ion transporter in that particular segment. And so I'd like to tell you a quick vignette story to kind of illustrate this theme on the first one on this list, olfactory receptor 1393 in the proximal tubule. And the work that I'm going to talk about here was all done by Blythe Shepard when she was a postdoc in our group. She's now an associate professor at Georgetown. But when Blythe started in the group, she wanted to understand what olfactory receptor 1393 might do in the kidney. And the first thing she did is that she tried to localize it. And she found that it's localized to the apical membrane of the renal proximal tubule. So let me tell you a little bit about the renal proximal tubule. So this structure right here is the glomerulus, the renal glomerulus, different than olfactory glomerulus, clearly, where the blood is filtered into what will become the forming urine, and then it travels through this tubule. And the very, very first segment it interacts with is called the proximal tubule, and that's where Blythe found expression of this olfactory receptor. The proximal tubule is famous for reabsorbing a lot of different things, but one of the very important things that it reabsorbs is glucose. And nearly 100% of the glucose that is initially filtered in our blood gets recovered completely back out of the urine back into the blood. This is because glucose is a precious substance that we used ATP to make. We don't want to pee it out in the urine, right? So the first thing that happens is it gets reabsorbed back into the blood. We made 1393 knockout animals, and Blythe found that they had a really curious phenotype, which is euglycemic glycosuria. So what I mean by that is normally, so glycosuria means that there's glucose in the urine, and normally that only happens if your blood glucose is high, right? So in uncontrolled diabetes, your blood glucose is so high that some of it, quote, spills over into the urine. But these mice had euglycemic glycosuria, which means that if you look on this axis, this is their blood glucose. There's no difference between wild type and knockout blood glucose. So it's not that they're hyperglycemic, but despite the fact that they have normal blood glucose, they're spilling glucose into the urine, as you can see on this axis. So that was really curious to us because typically that's indicative of a problem with the function of one of these glucose transporters that's supposed to reabsorb glucose from the proximal tubule back into the blood, called the sodium glucose cotransporters 1 and 2. Indeed, Blythe went on to show that in the absence of 1393, the membrane expression of those sodium glucose cotransporters, the SGLTs 1 and 2, was reduced. So she had this general idea that 1393 is acting to somehow support the activity and or expression of SGLT 1 and 2. And in line with that, Blythe went on to show that in both type 1 and type 2 diabetes models, the knockout mice actually do a little bit better because they're leaking some glucose into their urine at baseline. They can actually tolerate higher glucose a little bit more easily. And another thing that often happens to the kidney early in diabetes is called hyperfiltration, where the glomerular filtration rate is not properly regulated, and that's thought to be tied to how the kidney handles glucose. And it turns out that hyperfiltration was also attenuated in the knockout mice. So that was really intriguing to us. I want to leave this topic with a slide about why you should care about the things that I just told you. And so normally more often I'm talking to my fellow renal physiologists. In that case, my sell to them is, well, you should care about this because SGLT2 inhibitors are a really important clinical drug, right? So these have been on the market for a little bit over 10 years now. They're originally developed for type 2 diabetes, but now we know that very unexpectedly, they're also really efficacious for non-diabetic chronic kidney disease as well as for heart disease. And if our receptor is modulating SGLT, then maybe that's important. Maybe there's something better we can understand about SGLT2's function or about how SGLT2 inhibitors work. But for this crowd, I also, I think would be remiss if I didn't mention that SGLT1 has also been reported to play a role in the sweet taste pathway by some folks in this room. And so if you think about it that way, this is even weirder. You have an olfactory receptor that in one way is regulating a taste receptor in the kidney, which is just all kind of fun to think about. So I hope I've shown you, I've illustrated an example of this first theme, that novel receptors and the tubular modulating transporters that we already know are important in renal physiology. For the vast majority of my time, though, I want to talk about GPCRs in the renal vasculature. And we've studied 3 different GPCRs in the renal vasculature to date. And before I move on to focus on one of them, I just want to point out something that I think is weird and wonderful, if you will. So the 3 we've studied are shown on this slide. There's 2 olfactory receptors and 1 GPR. All of them are highly conserved across placental mammals. And the part is that they have really, really similar ligands. So all 3 of them are activated by a different subset of short-chain fatty acids, which have been mentioned in earlier talks today. So these are metabolites that are produced by our gut microbiota. So that is something else our lab has got really interested in, is how does the gut microbiota influence kidney and cardiovascular function? But for the purpose of today's talk, I'm going to narrow in and talk about the 3rd one on this slide, olfactory receptor 558. So I've reminded you that olfactory receptors are the largest gene family in the entire genome. And then I just told you for the rest of my talk, I'm only going to talk about one of them, right? So I want to give you a slide for why you should be okay with that and not be too sad that I'm not talking about the other 300 and some in humans. Or the other 999 some in mice. So one reason that I think you should be okay with me focusing on this receptor is that although there are a lot of different olfactory receptors, there's only 3 that have really clear orthologs across all placental mammals, and this is one of those 3. So evolution thinks that this receptor is important. I want to point out that the nomenclature for olfactory receptors is a hot mess sometimes, and that it's different in different species. The murine receptor is called olfactory receptor 558. The human ortholog is called OR51E1. I'm going to be talking about both of those receptors today, so I just wanted to try to introduce that confusion a little bit here initially. Interestingly, the human receptor is also one of the most well-conserved evolutionarily amongst the human olfactory receptors. So again, indicating that evolution seems to think that this receptor is important. So maybe we should figure out what it does. I am trained as a renal physiologist, and both the mouse and the human receptor are known to be expressed in the kidney. So if it's evolutionarily conserved and is in the kidney, like I'm already pretty interested, right? And then our lab has previously studied this receptor in vitro, and we were able to identify ligands for it. And I've already kind of hinted at this on the previous slide, but the best ligand for this receptor is a short-chain fatty acid butyrate. It does have a number of other ligands. The majority of them are microbial metabolites as well. So when we began to study this, our approach was first to try to localize the receptor because the different cell types in the kidney play very specific roles. So knowing the cell type is really useful in thinking about physiology. And then secondly, we wanted to know what it was doing there. And the work that I'm going to tell you about today is a work of a fantastic then postdoctoral fellow, now research associate in my lab, Jiajia Xu. When Jiajia was a graduate student at Hong Kong, she actually was a visiting student at Monell for some time. So she also has very fond memories of being around here as well. So when Zhao Zhao started working on this project, she wanted to localize this receptor, and very annoyingly, we've not been able to validate any antibodies in the field. We've tried to make our own as well, and olfactory receptors are just not very antigenic, and that's very frustrating. But Zhao Zhao was able to use RNA-scope, which I hope projects okay, in order to localize the RNA for this receptor. So on the top panel, you're looking at the green is standing for smooth muscle actin, which is a marker for vascular smooth muscle cells, which is a marker for blood vessels. And then the purple dots are the messenger RNA for our olfactory receptor. And I hope you can see that the purple dots are in the cells that are also marked green. The bottom panel is tissue from a knockout mouse that we made. And you can see that happily the signal goes away in the knockout. So there's localization to vascular smooth muscle cells in blood vessels. That's great. If you're a kidney person, then you might know that there are certain blood vessels in the kidney that are much more famous than others, and so we really wanted to look at those. So I'm going to orient you quickly here, just in case you don't stare at slides with sections of kidneys on them every day. If you don't, you really should, because it's pretty cool. But if you don't, let me orient you a little bit. So this thing that's labeled with a G in this drawing is the renal glomerulus. That's a tuft of capillaries where the blood is filtered into what will become the forming urine. And blood enters the glomerulus through this vessel called the afferent arteriole, and it exits through the efferent arteriole. So the afferent and efferent arterioles are particularly famous and important in the kidney. The afferent especially so because it's surrounded by these juxtaglomerular cells which store and secrete this hormone called renin. So renin is a hormone that is a rate-limiting step in the main pathway that controls our blood pressure, and that this is where that hormone comes from. So if you see staining in the afferent arteriole, I automatically start thinking about blood pressure regulation. And sure enough, this is showing you that there's a glomerulus in both wild types and knockouts. On either side of the glomerulus, there are two vessels that are the afferent and the efferent, and both of them in the wild type animals stain for olfactory receptor 558. We don't see any staining in the knockout. And just to make sure that this is in renin cells, we also co-stain for 558 with a renin antibody. And again, we see co-localization in the wild type, but not in the knockout. Now, a great thing about living in the year 2025 or 2023, actually, when we did this, is that if you want to know where something is expressed in the human, you can take advantage of these online databases. So we looked at these online RNA-seq databases to see if the localization we saw in the mouse was conserved in humans or not. And in the Kidney Tissue Atlas database, they identified 57 different cell clusters of different cell types. And the human receptor, the human ortholog, was expressed in 3 of those. 2 of them are vascular smooth muscle cell types, and the third one are those renin-positive juxtaglomerular cells. So that was really encouraging to us because that's the same localization that we saw in mice. Now, of course, vascular smooth muscle cells and blood vessels are not only found in the kidney, they're found in lots of other places. So a question that Zhao Zhao initially had was, wait a minute, is this receptor in blood vessels everywhere? Is it only in blood vessels in the kidney? So she wanted to look at that. And initially, she did a qPCR experiment and looked at a number of different tissues. And you can see that there's a range of expression. It's certainly not restricted to the kidney. Interestingly, there are some places where it's truly negative. We never see any expression in the cecum or the liver, but there's also places that are expressed at a much higher level than the kidney, like the heart and the aorta. We then wanted to see if when it's expressed in these different tissues, if it's always found in blood vessels or if it's found in other places. Oh, and this did not import properly. So I will just tell you, because my table isn't going to work, that it's only expressed in blood vessels and in all of the different places that we've looked. So sorry about that. That something didn't work there. So again, we took advantage of these online RNA-seq databases to see if for the human OR, if it had a similar localization, and in fact it does. So in heart and vascular smooth muscle and in adipose tissues, we primarily see localization to these smooth muscle cells, which aligns really well with the tissues where we see localization in the mouse. So it's encouraging to us at this point to go on and look at the physiological role, knowing that at least the localization is conserved between these species. So this is a question we really wanted to answer, right? What is a physiological role of the receptor? And just based on the localization alone, our hypothesis was that it might play a role in blood pressure regulation. Blood vessels are important in blood pressure regulation, and those renin-positive cells are very famous for their role in blood pressure regulation. So we wanted to measure We wanted to measure blood pressure in our wild type and knockout animals. And before I show you those data, I need to remind you of an important fact that you might not think about a lot. And that's the fact that there are sex differences in blood pressure, even in normal tension. So even in healthy folks, there are some pretty decent sex differences in blood pressure. So we're going to be focusing for this talk on the premenopausal years or the reproductive years, which I've put a little box around here. And you can see that during the premenopausal years, men have higher blood pressure than women by about 10 millimeters of mercury. So this isn't a small amount. When your doctor tells you your blood pressure, he tells you two different numbers. Both of those numbers on average are lower by about 10 in women than they are in men during these premenopausal years. So we wanted to measure blood pressure, but we knew that we would need to do it in both men and in both male and female mice because we knew that the wild types should have different blood pressures in those two different sexes. Luckily, not only do women have lower blood pressure than men, but this is modeled really well in the mouse models that we're using, where premenopausal females also have blood pressure that's lower than the males by a similar amount. So if you're not familiar, the way that we measure blood pressure, the gold standard way to measure blood pressure in rodents is using a technique called telemetry. So I'll just really briefly describe it. So telemetry requires a surgery where the tip of the catheter is inserted through the carotid, such that the very tip of the catheter is in the free-flowing blood of the aortic arch. This catheter is then connected to a transmitter that's tucked into the body of the mouse. And then they're sewn up and allowed to recover from the surgery for a couple weeks. And then we can turn the transmitter on remotely, and it sends a signal to a receiver in the same mouse room, much like the Bluetooth signal from your phone. And this allows us to measure blood pressure in these mice 24 hours a day when they're not restrained, they're not under any anesthesia, they're freely moving around the cage, living their little mouse life, and we can measure blood pressure and heart rate in real time. And so this is what that data looks like. So right now, just to orient you, I'm only showing you the data from the wild type animals. So looking across the graph, you're looking at mean arterial pressure, systolic and diastolic. So when your doctor tells you your blood pressure, the top number is systolic and the bottom number is diastolic. So blood pressure readings are over the 24-hour day. So this gray part here is when the lights are turned off in the room. Mice are nocturnal, so when the lights are off in the room, they're a little bit more active. So you can kind of appreciate that in the circadian rhythm of their blood pressure, that it's a little bit higher at night. One thing I hope that you'll notice on this graph is the fact that the black line is always above the blue line, which you can predict from the slides I just told you, right? That males have higher blood pressure than females, so the males in black have higher blood pressure than the females in blue. Now, at the same time that Jaja was doing these measurements, she was also measuring blood pressure from littermate knockouts from these same animals. And what she found really surprised me. What she found was that the sex difference disappears in the knockout animals. So in the knockouts, there's no longer any difference between the males and the females when we look at blood pressure for mean arterial pressure, for systolic blood pressure, or for diastolic blood pressure. Now, if you're like me, you're probably staring at this graph and you're trying to figure out if the sex difference went away because the males came down or because the females went up. And I can make that a little bit easier for you by taking the same exact data and plotting it in a different way on the next slide. So here, we're taking the same data, I've just plotted it differently. So again, you're looking at mean, systolic, and then diastolic blood pressure. And these first two bars are comparing male versus female in the wild type. And I hope you can appreciate that there's a sex difference there, right? The males are quite a bit higher than the females. And then the next two bars are looking at male versus female in the knockout. You can see the sex difference is completely gone. And really surprisingly to me, the sex difference is gone mostly because the females increase, but also because the males decrease. They kind of meet in the middle, which is really surprising. And one of the first questions that Jajo and I had when we initially saw this data was, wait a minute, are other sex differences intact, right? Did we break something bigger by knocking out this one receptor? So we looked at that, and as far as we can tell, other sex differences are intact. So if we look at simple things like body weight, data that we'd already collected because we weigh the mice before the surgeries, we could see that males weigh more than females, and both in the wild types and the knockouts. We also did a very, very simple quote unquote experiment where we put a male mouse in a cage with a male knockout in a cage with a female knockout. And we waited 3 weeks and we came back and there was pups in the cage. So we know that on a very, very simple level, both the knockout males and the knockout females must be fertile, right? We're going back now to try to look in a lot more detail about, pup number and pup size and all of that. But on a super, super simple level, we know that the knockout males and the knockout females are fertile. And so here's another chart that didn't import properly, but what this is supposed to tell you is that when we, I'll take it away because it's annoying. What that's supposed to tell you is that at this point in time, we also realized that when we initially looked at all the different tissues earlier in the talk to see where it's expressed, we kind of forgot about things like ovaries and testes. So we went back and looked at those. And it turns out that the receptor is expressed in blood vessels in both of those places, but not really anywhere else. So it's not in Sertoli cells or Leydig cells or anything like that. We really only see it in blood vessels. All right, so before I go on to talk a little bit more about how we're thinking about how we move forward with the story, I just want to give you a slide on the implications of sex differences in blood pressure, because to be honest, this is something that I didn't really think about before we stumbled on this pretty accidentally. So I mentioned to you earlier that premenopausal women have lower blood pressure than men do. But I think it's worth noting that the clinical definition of hypertension right now, at least, and this is being thought about, but right now the clinical definition of hypertension is identical in men and women. And that means that a premenopausal woman has to increase her blood pressure by 10 millimeters of mercury more than a man does before she's going to be diagnosed as hypertensive and then treated for hypertension in the clinic. And I think that's especially worth thinking about in light of a paper that came out at the end of 2021 in Circulation that showed that women actually incur increases in cardiovascular risk at much lower blood pressures than men do. So men have a higher absolute risk of cardiovascular disease. However, men don't incur increases in risk until their blood pressure increases quite a lot, whereas even very small increases in blood pressure increase risk for women. So I have two examples here. So compared to a reference value of a systolic blood pressure less than 100, women incur an increase in stroke risk once their systolic is above 120, and men have to have an increased systolic above 140 before they would have an increase in risk. I'd also refer you up to the clinical definition of hypertension. In these two examples, the man would be treated as hypertensive and the woman wouldn't be, even though their stroke risk is actually pretty similar. For an MI, which of course is a sciencey word for a heart attack, this is even a bigger difference. So women incur an increase in risk once their blood pressure, their systolic has gone up by between 10 to 20 points, whereas men have to have an increase in systolic of more than 60 points before they have a comparable increase in risk for a heart attack. So what this got us really thinking about is the fact that one reason I think that we treat men and women the same in the clinic is because as basic scientists, we've never understood why blood pressure is regulated differently in health in men and women or in males and females. And so that's something where we thought maybe our receptor very unexpectedly can help us to better understand how this receptor is supposed to work and how blood pressure is regulated differently in baseline in men and women. And maybe that can give us insights into how we can think about treating hypertension with a little bit more of a nuanced approach in the clinic. So with that in mind, we wanted to understand the phenotype a little bit better. And I'm just going to show you a couple little more insights here. Because of where it's localized, we thought that it must be either affecting that hormone renin or it must be affecting vascular function. So we wanted to look at those two things. And I'm going to show you a slide or two about renin and then a slide or two about vascular function. So first we looked at renin. And of course, we wanted to look at both males and females given the phenotype that we had. This first graph I'm showing you is just showing you that we know how to do qPCR. So here we're doing qPCR for 558 itself in wild type and knockout mice. And as you would expect, it goes away in the knockouts in both cases. Then I'll show you the female data first. In the females, we wanted to look at what happened to renin, to that hormone that's so important in blood pressure control. So we looked at renin messenger RNA by qPCR. We looked at glomeruli and counted how often they had adjacent renin stain. And the gold standard is to measure plasma renin activity, which is what is shown in E. And by all of these measures, we didn't see any differences. So it didn't look like in the female knockouts anything was happening to renin. However, in the male knockouts, what we saw is that all of these things were decreased. So in the males, there was less renin messenger RNA in the kidney. Their glomeruli were less likely to have adjacent renin stain. And on two different diets, the male mice had lower plasma renin activity. So this implies to us that this could be playing a role in the phenotype. To remind you, the males had lower pressure, especially for diastolic blood pressure, and a decrease in renin would be predicted to decrease blood pressure. So that seemed to align for us that this is a potential cause of that phenotype. Now, normally in the literature, when there's a change in renin, it's not because there's a change in renin content per cell, it's because there's a change in the number of renin cells. So I won't belabor this too much, but Jiazhou sat at the microscope and counted a lot of different cells to see if this was the case. And if you just look down here to B, what she found was that when you look at renin-only cells, that there is a decrease in the number in the male knockouts compared to the male wild types. We also looked at something called AKR1B7, which is thought to be a very, very cell-specific marker for these renin cells. And we saw that this renin cell marker was down in males, but not in females, indicating, again, that there are likely less renin cells in the male knockouts. So we think that a decrease in renin could help explain the male phenotype. What's going on with the females? So we measured a lot of different measurements of vascular function where we saw either no change or very, very slight and subtle changes, and I'm not going to bore you with. But I think the most interesting thing that Jiazhou found is when she measured something called pulse wave velocity. So let me briefly explain what pulse wave velocity is. So pulse wave velocity is a measure of arterial stiffness. So in general, you don't want your arteries to be stiff. You want them to be very compliant. So I've always been taught to think of this like a rubber band. So you want your artery to be like a brand new rubber band where you can stretch it and it can adapt to accept that increased load that comes with each beating of the heart during systole. However, arterial stiffness is often said you could think of as a rubber band that has been sitting in your kitchen drawer for way too long that you kind of forgot about, and you pull it out and it turns out to be very brittle and not at all compliant. And so that is what happens in aging. And arterial stiffness, in a word, is thought to be bad and to lead to higher blood pressure. So we wanted to measure arterial stiffness. We've since done this in the male knockouts. Right now, I'm going to show you data from the female knockouts. The way you do this is by putting a Doppler probe at 2 different points on the aorta in a live mouse under anesthesia. And you literally measure the distance that the pulse wave travels and the time that it takes. And so that's the velocity, right? So the pulse wave velocity is just the time that it takes a pulse wave to get from point A to point B. In a stiffer vessel, it's going to move faster. And indeed, the knockouts have stiffer vessels as evidenced by the fact that they have higher pulse wave velocity in this assay. So an increase in pulse wave velocity is expected to increase blood pressure. So we think that this increased arterial stiffness is likely a contributor to the reason that the knockout females have higher blood pressure. So I've focused a lot on olfactory receptor 558, but I want to come back a little bit to OR51E1, that human receptor that I had mentioned a little bit earlier in the talk. And around the time that we were kind of starting to finish up the story, Jiazhou and I got really excited when we dug through the literature and we saw this Nature Genetics article that had come out a few years before that showed that there is a human variant in OR51E1 that's associated with diastolic blood pressure in humans. So that was really encouraging to us. And we read this paper really carefully and we realized that they looked at both men and women, but they didn't do any analyses separately for men and women. So I sent the authors some annoying emails and asked them if they would collaborate with us. And luckily they were very kind and very agreeable to do that. So I'm going to show you next data for 2 different variants. So the first variant I'm going to show you for is a variant that they initially found in this paper. And the variant they initially found in this paper, again, they looked at males and females together. So it's not too surprising that when they split males and females separately, they kind of see something similar, which is why it was significant to come up in their initial analysis. But just to walk you through this here briefly, I should say it's a known phenomenon in blood pressure research that common variants have infinitesimally small effects on blood pressure, whereas rare variants tend to have much larger effects. So this is a common variant. About 21% of us are carriers to the minor allele of this variant. And indeed, it has an infinitesimally small effect on blood pressure. So these betas are in millimeters of mercury. So this is, less than 0.2 millimeter mercury change. So it's a very small change, statistically significant only because the N is very large from the UK Biobank. And not too surprisingly, given that it came up in the initial analysis, when you look at males and females separately, you see something pretty similar, but still interesting that it does affect blood pressure. The more interesting variant, I think, is a variant that we also asked these authors if they could look at, because we had studied this variant previously in an in vitro study of this receptor, and we'd gotten interested in it. So we asked them if they would take a look at it. And here the story got a lot more interesting. So if you look at all, which is males and females together, there's no change in blood pressure, which is why it didn't come up in their initial analysis. But when you split out males and females, you can see that in males, this is a very rare variant. This rare variant decreases blood pressure in males by almost 5 millimeters of mercury and increases blood pressure in females by almost 6 millimeters of mercury. So it was really exciting to us because although this is a very rare variant, this is the same magnitude and direction approximately as what we see in the knockout animals. So this is really, really intriguing to us. In fact, if you look down here at the graph for sex, you can see that in the control genotype, the males have higher blood pressure than the females, but this actually switches in people with the minor allele of this variant. So I'm gonna conclude this part of the talk and then just give you some general thoughts, and then hopefully maybe even get us back on time. So I hope I've shown you that this receptor is an evolutionarily conserved olfactory receptor that's required for sex differences in blood pressure. In females, we think it acts by altering arterial stiffness, whereas in males, we think it acts by altering renin. And I want to spend my last couple of minutes just telling you kind of how we're thinking about this receptor going forward. And honestly, I'd love ideas because we're definitely still figuring out the best way to approach this. But one question that keeps me up at night, if you will, is how is it that the receptor functions so differently in males and females? How is it that when you have a knockout, the effect is literally the opposite in a knockout female or a knockout male? So this is something we're trying to understand. And I'm going to tell you right now, I don't know the answer to this question, but we're really thinking a lot about the role of a potential role of sex hormones to somehow be regulating this receptor. And I just want to give you two really simple reasons why I think that is likely the case. So the first reason is the fact that if you remember this graph that I showed you earlier, men have lower blood pressure than women until menopause, right? And then it changes. And in fact, both men and women have big increases in blood pressure with aging, but women much, much more so, such that women are hypertensive relative to men and the aged. So the fact that this sex difference disappears at menopause or right around the menopausal transition really makes us think that that there's an important role for sex hormones here. And I'll also point out, I kind of skipped over this when we initially looked at this slide, but for females, our co-authors actually split this data into females below the age of 50 and above the age of 50 for this rare variant. And there's actually a much, much bigger effect in females below the age of 50 as a very rough cutoff for menopause than in females above the age of 50. So on the flip side of the aging spectrum, I guess the other easy rationale for why we should be thinking and why we are thinking about sex hormones is that one thing I haven't told you yet is that sex differences in blood pressure actually don't emerge until puberty. So boys and girls before the age of puberty don't have differences in blood pressure, but right around the age of puberty is where sex differences in blood pressure emerge. So for all of those reasons, we think that sex hormones play an important role in regulating this receptor, and we're doing a bunch of different things to try to better understand that. And I don't have a clear answer for you yet. So I'll leave you with a list of future directions, which is really, to be honest, a list of questions I don't know the answer to maybe save time. One question that I've already kind of presented to you is how is it that the receptor knows that it's in a male or a female in order to act so differently? And we think sex hormones are involved, but we're still piecing that out. We want to know what happened, given these changes that happen during the life cycle, we want to know what happens to this receptor and to knockout phenotypes during development, during puberty, during aging, and also during pregnancy. So this is an interesting thing to think about, but nobody really knows why women have lower blood pressure than men do. But most people in the field, if you ask them why, they'll wave their hands and they'll say, well, it has something to do with the cardiovascular adaptations to pregnancy that have to happen in the woman, right? So maternal cardiovascular adaptations to pregnancy are really dramatic and really huge. And so we suspect that our receptor may also be playing a role in that. And we have some early data implying that it is. So that's something we're excited to look at more as well. I told you at the beginning that this receptor is activated by metabolites that are made by the gut microbiota, and then I kind of just dropped that thread, but that is something that we're still thinking about, and that I think is potentially really exciting. This receptor also has more than one ligand, and so another thing that we think about a lot is which of these ligands is actually important in vivo, or is it the conglomeration of all the ligands that's important in vivo? I told you earlier that my bent is to think about physiology, but there's also big sex differences in hypertension. And that's something that we also want to look at with women being relatively protected from hypertension until menopause. And then we have a great pharmacology collaborator who's developed some really novel synthetic ligands, both agonists and antagonists for this receptor, which is really helpful because the natural ligands activate our receptor and like 42 other things. So it's really useful to have these synthetic ligands that are much more specific. And then finally, we are also interested in looking at taste receptors. And I'll say that Jajel, the research associate in the lab that I mentioned earlier, is actually on a job interview today for a faculty position, and she is planning to have a lab that focuses on trying to understand the role of taste receptors in the kidney. So maybe at some future Monell symposium, she can come back and talk to you guys all about that. And with that, I need to thank everybody in the lab who actually did all the work that I talked to you about. Of course, we're very grateful for the funding that allows us to do any of this at all. And with that, I will thank you for your time and for the invitation. And if time allows, I'm glad to take questions.
Fantastic.
Thanks.
There is time for questions.
I have a question that was really interesting. I wonder if you know the transduction link from the receptor to glucose transport and from the receptor to reading cells.
Yeah, we don't in either case. We have good guesses. We don't have in vitro cell models that we trust for either of those, and teasing it out in vitro has been a lot harder than I had hoped.
That might be really interesting.
Yeah, for sure. So cyclic AMP is known to be involved in renin, for example. Like there's easy connections one can make from the literature, but we haven't shown that ourselves.
PNJ to sex linked.
Yes.
So those are the people who talked about taste, and you got to taste right at the very end. Obviously, the renin-angiotensin system is involved in salt handling. Do you have any reason to think that salt is involved somehow in this receptor activity?
Yeah, it's a good question. I can't completely rule it out, but we did put these mice on a high salt diet, which is a common intervention to try to increase blood pressure if there's salt sensitive blood pressure. And they didn't change blood pressure any more than the wild types did. So that didn't uncover anything. As far as we could tell, they ate the same amount of diet on high salt as the wild types and the knockouts. So that it's a great thought. I can't completely refute it, but I don't have any data to support it. Yeah.
Thank you very much. You talked about the potential ligands at the end, and you also said that butyrate so far is the best ligand that is identified. Can you tell something about the range which you need to activate the receptor? So are plasma butyrate levels sufficiently high enough to activate this receptor?
Yeah, very good question. So plasma butyrate levels are right about at the activation threshold of the receptor, but far below the EC50 of the receptor. But they are at the range where we can see activation, like in a cyclic AMP assay in vitro. So we've been thinking that maybe it's more of an on-off thing instead of a maximally activated thing or not. But yeah, those levels are right at the place where they might be tickling the activation of the receptor.
So you talk a lot about this as a kidney receptor or related to the kidney and related to blood pressure for that reason, but it's also expressed in other places. I'm curious if you do a tissue-specific knockout in the kidney. Do you see this effect, or could it be mediated by some other expression in some other part of it?
Yeah, great question. So we haven't, I can't answer your question yet, but the knockout that we have, we can't do a tissue-specific knockout, but we can do a tissue-specific rescue. So we've done some initial experiments where we did a tissue-specific rescue in the kidney, but we're still phenotyping those guys. Good question.
Hi, Jennifer. Thank you very much. Today we are honoring to Bob's retirement and service system model in science, but the topic you mentioned here and also the postdoc you mentioned, she's Jessica. We used to call Jessica Jan.
Yes.
She was my PhD student, and it is very nice to hear after many years.
She's fantastic.
Jessica is did a great job in your lab.
Absolutely.
And I have a couple of questions. I don't know. In the very first slide, you mentioned that R58 or
Mhm.
51E1 also related to the NHE3.
Related to,
NHE3.
I think that was it. So there's a different receptor that's related to NHE3. I think I might have confused you. So we have studied a GPCR that's related to NHE3. Yes, C5C, yes.
51E1, we work extensively, is a pretty large ligand family. You are right, the butyric is a ketogenic amino acid product, ketone product.
Yeah.
Which is related to the diabetes or is a sort of metabolic marker.
Yeah.
And also R51 is accepted as a molecular receptor, which covers a lot of any odor which reaches certain concentration, activate the R51E1.
So I think I followed most of that, but I missed the question at the end. Can you?
The question is that the R51E1 is, how do you determine it is specific for the renal productions?
So for the human receptor?
Yes.
Yeah. I mean, so for the human receptor, we only have indirect evidence, right? It's expressed in the right cell. It's expressed in the renin-containing cell, and then in the mouse, we see changes in renin. So we assume that the human receptor might be doing the same thing, but we've not measured renin in humans.
Okay, thank you very much.
Yeah.
A quick question, is this receptor expressed in olfactory tissue?
Yes, it is. Zone 1 and zone 2 mostly.
That was a really nice talk. I have a question, actually, sort of two short questions having to do with the anecdote you had at the beginning. So for the olfactory receptor 1393, you said that it was involved in glucose reuptake. But this is the mouse receptor. Do you have evidence that that's the same thing that's going on in humans?
We've not, I mean, Blythe took that project with her, so I might be missing something, but at least when she was in my lab, we did not look at we looked to make sure that the ligands were the same. No, we didn't even do that. So no, we don't have evidence for that. In fact, I would say that the human ortholog for that one is a little bit touchy because there's more than one human receptor that has a really similar sequence similarity. So I don't think we're entirely sure which human receptor to look for.
Yeah, I mean, and sort of a follow-up to that, if the human biology was similar, is that if you think about a lot of cancers, of course, are driven by glycolysis.
Yeah.
And so you can imagine, it would be interesting to see whether this type of biology is driving some kidney cancers, or it becomes a driver.
Yeah, I actually haven't thought of that. It's a great point. Thank you.
There's a tiny question, brief question here. Maybe we
You
could.
can shout and I can repeat
You
it.
can repeat it.
Or you can come up.
So if you stratify the UK biobank and the rare SNP by women who are or are not on hormone replacement therapy, do you still see the effect?
So the question is, if you stratify the UK biobank data by women that are on human hormone replacement therapy or not, do you still see the effect? I don't know the answer to that. I didn't think to ask that question. But it would be really interesting to see if they have that data, if they can do that. It's a great idea.
All right. Thank you very much.
Thank you.
That was fantastic.